So we're going to talk about both prokaryotes and eukaryotes in this chapter. And first we'll go over some of the main differences between them. The defining difference between prokaryotes and eukaryotes is that prokaryotes have a nucleus and eukaryotes do not. Actually, prokaryote means before nucleus and eukaryote means true nucleus. You have two domains of life that are prokaryotes, bacteria and archaea, and eukarya are the eukaryotes. You have microbes of both types, of course, in all three domains. In addition to not having a nucleus, prokaryotes don't have other membrane-bound organelles in general. There are just a few exceptions of prokaryotes that have membranes inside of them and even something that could be called an organelle. But eukaryotes always have a lot of membrane-bound organelles, so they're highly structured and highly organized. They're also larger cells than prokaryotes. But despite all those differences, the defining difference is that a prokaryote does not have a nucleus and a eukaryote does have a nucleus. So here you have kind of a basic diagram of a prokaryotic cell and a eukaryotic cell. This is not to scale, so the prokaryote is shown as being about as big as the eukaryote, but if you look at the scale bar, you can see that there is actually a big difference in their size. So the prokaryote would be just really tiny compared to the eukaryote. The prokaryotic cell has stuff in it, as you can see, but it's not organized by membranes. When you compare that to the eukaryotic cell has all these sheets of membranes inside of it. Those are its membrane-bound organelles, so it's a much more organized structure. In the prokaryotic cell, you do have some organization, but mostly stuff is just kind of floating around in there. So first we're going to go through the prokaryotes, which are bacteria and archaea, and we'll talk about the structures and the basic function of those structures in those types of cells. So first, we'll talk about morphology. Morphology just means shape, and you have a variety of shapes that prokaryotes can come in. The most common two shapes are coccus and bacillus. Coccus is just a ball or a sphere, and a bacillus is a rod or a cylinder. Sometimes they're actually just called rods. That's pretty common, actually. The plural for coccus is cocci, and the plural for bacillus is bacilli. So most bacteria and archaea are either a coccus or a bacillus. But then you also have a lot of other shapes that they can take as well. So you have the coccobacillus, which is kind of midway between a sphere and a rod. So in the image here, you can see a coccus is just a sphere, the bacillus is a rod, and the coccobacillus is it's more like two spheres stuck together, kind of. It's like too big to be a sphere, but it's not really long enough to be a proper rod. Then you have Vibrio. Those are shaped like a boomerang, basically. You have Spirillum, which is a loose spiral shape. And you have Spirochete, which is a tight spiral shape, more like a corkscrew. So those are similar, but the spiral is tighter in the spirochete. You also have pleomorphic microbes that kind of have just not really a well-defined shape, and the cell can take different shapes throughout its life, and none of them are really looking like anything specific. So we just call that pleomorphic. That means many forms. Then you also have a lot of rare forms, especially in archaea. A lot of them have unusual forms like stars, triangles, rectangles. You have some microbes that have what's called a budding or appendage shape, where there's like part of the cell that's more bulbous and then a thin part sticking out from that. And then you have filamentous microbes, where it's a really long hair-like cell, basically like a filament. And then you also have different cell arrangements that are caused by the way that different species reproduce. So a lot of bacteria and archaea will only divide in certain planes when they reproduce. So they'll only divide like side to side, or they'll do side to side and top to bottom. And then if they remain attached to each other after they've divided, then you have an arrangement. So there's several arrangements that are common to find for cocci. First you have diplococci, just 2 cells stuck together. Then streptococci, which is a long chain of cells. You can find tetrads, which are 4 cells arranged together like a square. You can also find sarcinae, which are more like 8 cells together in a cube. And then finally, Staphylococcus. Staphylococci are just a random arrangement of the cells that are stuck together. In order to get the Staphylococcus pattern, you have to have cells that don't divide in any specific plane. They can do any plane. So the genus, there's also a genus of bacteria called Streptococcus and another one called Staphylococcus. So that can get confusing. Are we talking about the arrangement or about the genus? You just have to look at the way that the word is written. If it's capitalized and italicized, that's a genus name. If it's not capitalized and it's not italicized, then that's an arrangement. And you also have some common arrangements for bacilli. You have the diplobacilli, just 2 bacilli stuck together. Then you have streptobacilli, a long chain of bacilli. And they can form palisades and V-shapes as well. Palisades would be when the cells are kind of stacked next to each other, and some of them may be tilted. And in the V-shape, 2 cells are attached to each other at an angle that looks like a V. So there's a special type of cell division called snapping division that forms the palisades and the V-shapes, and we'll get into that later on in the semester. For some of the other cell types, like the spirals and the vibrios, they don't really have arrangements. You always find them as single cells, and that's just because they don't remain attached to each other after they've divided. So one of the features of prokaryotic cells is that they're very small compared to eukaryotic cells. So even if you have 2 cells that are both microbes, one of them is a eukaryote like a yeast and one of them is a prokaryote like a bacteria, the bacteria is going to be much smaller than the yeast cell in general. And there's some reasons why it's better to be smaller for a microbe. And mostly it's to do with the surface area to volume ratio, or the SV ratio, which is just a measure of how much surface area you have compared to how much volume you have. So when an object gets bigger, the volume goes up much more quickly than the surface area goes up. So bigger things will have a bigger volume compared to their surface area, and smaller things will have a smaller volume compared to their surface area. So if you look at these 2 spheres, one of them has a radius of 1 micrometer, the other one has a radius of 2 micrometers, but you can see that the one that has a radius of 2 micrometers has a very big volume compared to the 1 micrometer ball, and the surface area has not gone up. It has gone up a lot, but not as much as the volume has. So when you calculate the surface area and the volume for spheres with these radii, you get that the surface area compared to the volume for the first sphere is a ratio of 3. So that's 3 times as much surface area as volume. And for the second sphere, it's 1.5. So that's 1.5 times as much surface area as volume. The volume has gone up much faster than the surface area has in the bigger sphere. That's important because on the surface is where the cell is doing exchange with its environment. So if you're going to get nutrients from the environment, it has to come into the cell at some point on the surface of the cell. So the bigger the surface area the cell has, the more places it has where it can bring nutrients in, and the more places it has where it can get waste out of the cell. The volume of the cell, on the other hand, that's where all the stuff in the cell is going on. That's where you have your metabolism. So that's where the cell is using its energy. So the more volume you have in general, the more stuff you have going on in there that needs energy. So in general, a smaller cell is going to have an easier time getting nutrients and will not need as many nutrients, and a bigger cell is going to have a harder time getting nutrients and it will need more nutrients. So that means that the smaller cell is favored. The smaller cell is going to be able to grow faster. When the smaller cell is able to grow faster, it's going to be able to divide faster and reproduce faster. So that means that if you have a population of big cells and a population of small cells, the small cells are going to multiply more quickly and they're going to come to dominate the big cells. Since they're reproducing more quickly, that means that they're evolving more quickly also. So a small cell is always, well, almost always in general, is going to evolve more quickly than a big cell will. And when a cell is evolving, well, a single cell doesn't evolve, but a population of small cells will evolve more quickly than a population of big cells. So that means that those small cells are at an advantage if the environment changes. If the environment changes, they're going to be able to react to that more quickly and evolve more quickly to be adapted to the new environment better. The big cells, the population of big cells, are just going to take longer to do that. And so that's a disadvantage for them. There is, however, a limit to how small you can get away with being, because you need to have like all the stuff that you need. You need to have space for your genome, and space for your ribosomes, and space for your metabolism. So you can't just be so small that there's nothing to you at all, or else there's not enough there for a cell. Another reason why it's good to be small, well, it's kind of related. Prokaryotes will always evolve more quickly than eukaryotes. And part of that is because the prokaryotes are smaller than the eukaryotes are. But part of that is also the fact that prokaryotes have a haploid genome and eukaryotes have a diploid genome. A haploid genome means that there's one copy of every chromosome. A diploid genome means that there's two copies of every chromosome. In general, if you have a haploid genome, you're going to evolve faster because if you have a mutation, it's going to be expressed. In a diploid genome, you have two copies of every chromosome. That means you have two copies of every gene. So if you have a mutation to one copy, it doesn't necessarily get expressed in the final organism because you still have one original copy as well as one mutated copy for that gene. So that means you can have carriers for a trait, like for instance, for some genetic diseases in humans, you can be a carrier if one of your parents had the disease, but the other parent did not, which would mean that you're not expressing the mutation that causes that disease, but you still have it in your genome. So if you don't have the ability to carry traits genetically without expressing them, if everything that you have in your genes gets expressed because you only have one copy of that gene, you're going to evolve more quickly as a population. So now we'll go through the parts of a prokaryotic cell, starting with the cell membrane. Other words for the cell membrane are the plasma membrane or the cytoplasmic membrane. A small cell membrane has a different composition for different prokaryotes, for bacteria versus archaea, but it's always a double layer, well, actually I should say almost always a double layer of lipids. So here you have the top layer, or the external layer of lipids, and here you have the internal or bottom layer of lipids. Almost all prokaryotic cells have this double layer of lipids in the membrane. These are special lipids. A lipid is defined as an organic molecule that does not dissolve in water, but these lipids are special because they actually have part that will dissolve in water and part that won't. So they have a hydrophobic end and they have a hydrophilic end. And these balls at the top, the heads of these lipids, are the hydrophilic end. The tails are the hydrophobic end. So the hydrophobic tails are going to end up pointing towards each other, and you're not going to have any water in this space that they create. But you do have water inside the cell and in the environment. So the hydrophilic heads are always going to point outside to the environment and inside to the cell. So they're always going to be lining the surfaces of the membrane. And that way the surface of the membrane is able to interact with water, and the interior keeps water out. And that provides stability to the structure of the membrane and lets it act as a barrier. You can have different things attached to these lipids. They can have little bits of sugar and stuff attached to them, and you can have proteins embedded in the membrane. So all membranes actually are going to have a bunch of different proteins embedded in them, and in this diagram here you can see a selection of proteins that are stuck into this membrane. Some of them are sticking all the way through, some of them will only be like attached to one side or the other, but they're all associated with the membrane. If we're talking about bacterial cells, then the lipids that are making up that membrane are phospholipids. So in this diagram, this is a phospholipid. The tails are made of fatty acids, and the head is a glycerol with a phosphate and with something else, usually a choline, attached to it. So the phosphate group has negative charges on it. The glycerol has polar bonds. So this part, the head, is always able to interact with water. The tails only have nonpolar bonds and no charges, so they do not interact with water. They are hydrophobic. And in bacteria, you're always going to see those phospholipids in the membrane. They're also making up the membrane of your own cells, actually. In Archaea, it's different. You don't have phospholipids. You have a bunch of different types of lipids, actually. But what they all have in common is that they're going to have a hydrophobic end and a hydrophilic end. So the basic purpose of the cell membrane is to keep the cell separate from the environment. It's to keep all the stuff the cell needs inside of the cell and to keep things that would be dangerous to the cell outside of the cell. And it also has selective permeability, which means that it allows some things to cross but not all things. That way the cell can get nutrients into the cell through the membrane, and it can transport wastes from inside the cell out to the environment. Another benefit of the cell membrane for prokaryotes is that it allows energy to be stored in what's called the proton motive force. So the proton motive force is basically a concentration gradient for protons across the cell membrane. And the cell is going to be basically generating protons inside of it and then pumping them out to the environment. So outside of the cell, you end up with a lot of protons, and inside the cell, you have not very many protons because they're always being pumped out. So that creates a concentration gradient with a lot of protons outside and a few protons inside. And then you can let some of these protons move back across the membrane, and they're going to do that spontaneously down their concentration gradient, and the energy of their traveling can be used to power different processes. So that means that the membrane of the cell for a prokaryote is kind of like a battery. It's storing this proton motive force. You can use that proton motive force to make ATP, you can use it to move things across the membrane, and you can use it to rotate your flagella if you have them. Outside of the cell membrane, you usually are going to find a cell wall. Almost all prokaryotes are going to have a cell wall outside the cell membrane. Not literally all of them, but most of them. The purpose of the cell wall is to provide support and protection for the cell. So it gives it some kind of shape, basically, because the cell wall is a rigid structure, so it gives the cell its shape. And it also will protect it from the environment. Specifically, it's protecting it from osmotic pressure. So in general, cells are going to have more solutes inside of them than the environment that they're in. So if you have some cells floating in some water, there's going to be more solutes inside the cell than there are in that water that they're floating in. That means that they are in a hypotonic solution, and they are almost always in a hypotonic solution. It's just the way it goes. Anytime that you have a cell inside of a hypotonic solution, you're going to have water moving by osmosis to the inside of the cell where you have more solutes. Water will always follow the solutes. So if water is moving into the cell, then the cell's volume is getting bigger and bigger. And at some point, if you have too much water move into the cell will burst and die. So in order to prevent that from happening, prokaryotes have a cell wall. So the cell can get bigger and bigger until it's pushing up against the cell wall. And the cell wall is a very strong, rigid structure. So it can't get bigger than that, and it can't burst the cell wall. What's going to happen is you just don't have water moving into the cell anymore. So it's very, very important for bacteria and for Archaea that they have this intact cell wall that allows them to survive in hypotonic environments. On the other hand, if you have a hypertonic environment, the opposite thing would happen. You would have more solutes outside of the cell than inside, and water is going to be sucked out of the cell, following the solutes going to the environment. It's not very common to have a hypertonic environment. Most of them are hypotonic, but you do have some. And the cell wall does not protect the cell against a hypertonic environment. If the cell is in a hypertonic environment, it risks losing all of its water and shriveling up and dying. And this is the reason why you don't have to refrigerate honey, basically. You can just leave it out on the counter. It's just sugar. Bacteria would love to eat it, but it's hypertonic. It's got so much sugar in it that it's hypertonic, and they can't survive there. So making something hypertonic is one way to kind of preserve it. It's also important, the cell wall is also important for medical science, because if you're going to have an antimicrobial drug that is going to kill a prokaryote, it has to get through the cell wall to get into the cell, of course. And you have different types of cell walls. And so different types of drugs will be able to cross different types of cell walls. And if your drug does not cross a cell wall, it does not work against that type of prokaryote. Since this is medicine, we would just be talking about bacteria, because archaea don't cause disease. Also, some drugs target the cell wall specifically, mostly targeting the synthesis of the cell wall so that these bacteria cannot build new cell walls. If they're not able to build their cell wall, then they're going to die. Yeah, they need that. You have two basic types of cell walls in bacteria. You have gram-positive and gram-negative cell walls. Both of those types are made out of a polysaccharide called peptidoglycan. The glycan part means that it's a polysaccharide, and the peptido means that it has a little bit of protein or some amino acids stuck onto it. And so we'll go over those two types of cell walls right now. But first, a little bit more about the structure of peptidoglycan. So you don't need to remember the details of this structure actually, but you have two basic sugars that make up peptidoglycan, and they just repeat to form the long chain of peptidoglycan. First you have NAG, which is N-acetylglucosamine, and then you have also NAM, which is N-acetylmuramic acid. Both of them are like modified forms of glucose actually. They're attached to each other using beta-1,4 glycosidic linkages. So this is a diagram of sort of this basic subunit that makes up peptidoglycan. So here's your NAM sugar, here's your NAG sugar, and then they're attached to each other right here with a beta-1,4 glycosidic linkage. It's important that it's a beta-1,4 glycosidic linkage as opposed to an alpha-1,4 glycosidic linkage because you don't have enzymes that can degrade the beta-1,4 glycosidic linkage. You have enzymes that can degrade the alpha-1,4 linkage, but not the beta-1,4 linkage. So that makes peptidoglycan like pretty stable. A lot of organisms don't have an enzyme that would be able to degrade it. It also makes the chains straight. If you have an alpha-1,4 linkage, the chain overall is going to spiral. If it's a beta-1,4 linkage, it makes a straight chain. So then you get these long straight chains of peptidoglycan. And also the NAM sugar has these few amino acids attached to it, and they're shown in this diagram in green. And wherever you have two chains of peptidoglycan that are close to each other, the amino acid bits are going to line up so that you can have hydrogen bonds forming between them. And that is what's giving the structure a lot of its strength. So you have these long chains of the peptidoglycan. They are straight so that they're able to stack right next to each other. And then you get hydrogen bonds between these amino acid add-ons. And all those hydrogen bonds, like if you have just one hydrogen bond, it's very weak. But if you have a lot, then it's very strong. So all these hydrogen bonds are creating a lot of strength in the structure, and they're really keeping these chains stuck together very well. And so all of that together will make the bulk of the cell wall. If you have a gram-positive bacteria, then the cell wall is basically just that peptidoglycan. If you have a gram-negative bacteria, then you have some more stuff. In red, you have thin layers of peptidoglycan here. Then you're going to have a space in blue and another membrane actually in green. And we'll go over the detailed structure for both those types of cell walls. So first, the gram-positive, which is the more simple type. It's basically just a lot of layers of the peptidoglycan, very thick. You also have some other molecules running through. You have these guys called teichoic acids that are linking the layers of peptidoglycan together. And then you have even bigger ones called lipoteichoic acids that are running through the whole cell wall and also going down into the cell membrane beneath it. So they're called lipoteichoic because they go down into the lipid part of the cell membrane, and they have hydrophobic parts right there that are going to anchor into the cell membrane. So they're basically keeping the cell wall attached to the cell membrane. So you're going to have your cell membrane, a small space, and then the cell wall made out of the peptidoglycan with these lipoteichoic acids anchoring the wall into the membrane. And then in the diagram, green you have the cell membrane, in blue you have a small space, and in red you have the peptidoglycan layers of the cell wall. The gram-negative cell wall is more complicated. You only have a few layers of peptidoglycan in it. So in this diagram here, first you have the cell membrane, then thin layers of peptidoglycan, and then you have another membrane, an outer membrane. And in between the peptidoglycan on either side, you also have a small space. So this outer membrane is it's different from the cell membrane in its structure. It's going to have different proteins in it. It also has this structure right here coming off of it, which is a molecule called LPS, which we'll actually talk more about on the next slide. The benefit of this membrane is that it keeps some things out that would be able to get through the gram-positive cell wall, specifically anything that's hydrophobic. Hydrophobic things can go through the peptidoglycan layers of the cell wall, but they don't go through that outer membrane, which is actually unusual. Most membranes would be permeable to something that's hydrophobic. The cell membrane is permeable to hydrophobic things, so they can just go right through the cell membrane. But the outer membrane, because of that LPS, it's not porous for hydrophobic substances. They can't get through. You also have a special name for the space between the cell membrane, and the outer membrane, which is called the periplasm. So that would include the space that's below the peptidoglycan and the space that is above it. Together, that's the periplasm. So the periplasm is special because you can it's like a place where the cell can stick stuff, and the stuff will not be inside the cell, but it's like close by and it can't just go off. So for instance, the protons for the proton motive force, if you have a gram-positive cell, those can kind of like diffuse off into the environment. In order to keep the gradient strong, you have to keep pumping protons out there. But if you're a gram-negative bacteria and you have your periplasm, then you pump the protons into the periplasm and they basically just stay there. So you're preserving the strength of that gradient by just having a place where you can stick things like that. So for that outer membrane of the gram-negative cell wall, I had mentioned you have this molecule called lipopolysaccharide in it, or LPS. LPS is a polysaccharide, and it has some lipid attached to it also, so that's why it's called lipopolysaccharide. And you actually have a lot of that LPS in the membrane. So the normal membrane would be almost all phospholipids plus some protein stuck in there, but on the outer membrane, it's about 50-50, like phospholipids and LPS. Here you have a larger diagram of the LPS. So you have the polysaccharide part, and then you have a lipid part called lipid A. The lipid part is going to be inside of the outer membrane, and the polysaccharide part is what is sticking up from it and making it all kind of fuzzy looking. The polysaccharide part has charges on it, and that is why the outer membrane cannot be penetrated by hydrophobic substances the way that a regular membrane could be. The lipid part, the lipid A part of the LPS, is actually toxic. So normally that's not a problem if you have an infection with gram-negative bacteria, since the lipid A part is embedded in the outer membrane, you're not actually exposed to it, and so it's like fine. But when the cell dies, when that bacteria dies, then its outer membrane is going to be degraded, and then the lipid A is going to be exposed to your body tissues. And it actually causes just an extreme reaction from the immune system, which causes a lot of inflammation. The inflammation can be very damaging if you have too much of it. If you have these bacteria like in your blood, and you take an antibiotic and they all kind of die like at one time, then all of a sudden you have a bunch of lipid A in your blood, and the immune system will react very strongly to that, and now you have inflammation in your blood and in your blood vessels. And if that is severe enough, you can actually enter shock and die. Inflammation will cause your blood pressure to go down. Well, it causes vasodilation, and if you have too much vasodilation, that causes your blood pressure to go down, and if you lose too much of your blood pressure, then you enter shock, which means you can't supply enough blood to your organs, and you will die. So for that reason, it may be important to know about LPS. Another word for lipid A is endotoxin. So if you're looking at kind of an older resource, a lot of the time you'll hear them talk, or you'll read them talking about endotoxin, and that just means the lipid A part of LPS. Not all gram-negative bacteria have the exact same LPS. They all have LPS, but they have different versions of it. Some versions are worse than others for you in terms of the toxicity for you. So some of them are not so bad, but some of them are pretty bad. Another special thing about this outer membrane is the porin proteins that you have in it. So some of these proteins that are embedded in it are called porins, and their basic job is to let stuff go through the membrane. So anything the cell needs, anything nutrient-wise that it needs is going to be able to pass through one of these porins to get into the periplasmic space, and then the cell can transport it across the cell membrane into the actual cell. And you also have this molecule called bronze lipoprotein, which is anchored into the bottom part of the outer membrane, and then it's anchored into the peptidoglycan also. So it's basically just keeping the outer membrane attached to the peptidoglycan. And that's the gram-negative cell wall. So archaeal cell walls are again different from bacterial cell walls. Most archaea do have a cell wall, and there's a lot more variety in their cell walls than what you have for bacteria. For bacteria, it's almost always gram-positive or gram-negative. There is like a couple rare types of cell walls in bacteria that are different, and a couple bacteria that don't even have a cell wall, but mostly it's gram-positive or gram-negative. In archaea, you have a lot of different types of cell walls that they can have, so we're not going to talk about them like with too much detail because it's just too much to cover. In general, they're going to have some type of polysaccharide making their cell wall, but there's a lot of different polysaccharides that they can use. One that's very common that they use is called pseudomurine. Murine means wall, and that's related to peptidoglycan, but it's a little bit different. So actually another word for peptidoglycan is murine, and another word for pseudomurine is pseudopeptidoglycan. Murine is kind of the older word, but for archaea, it's like pseudomurine is still more common to use than pseudopeptidoglycan, probably because it's like just shorter, is my guess. So here you have the basic subunit for peptidoglycan. You have your NAM sugar, your NAG sugar, the beta-1,4 glycosidic linkage, and then some amino acids coming off. For pseudomurine, it's similar. You have an NAG sugar, and then you have an NAT sugar, which is similar, but it's a little bit different. And you have a different bond between them. Instead of a beta-1,4 glycosidic linkage, it's a beta-1,3 glycosidic linkage. That's important because the enzyme that breaks the beta-1,4 linkage does not break the beta-1,3 linkage. You have to have a different, more specialized enzyme. And then again, you also have your amino acids attached to one of those sugars. So it's going to be structured in the same basic way where you have these long straight chains of pseudomurine, that are kind of stacked next to each other, and then you have these amino acid parts lining up with each other and hydrogen bonding to each other to give the structure some strength. Another common thing to see in the cell walls for archaea is the S layer. The S layer includes protein and glycoprotein. Glycoprotein is just a protein that has some sugar attached to it. And here you have an image of an S layer. So you have these different protein subunits in it, and then some of them would be glycoproteins, so they have sugar attached to them. Different archaea will have different sort of arrangements of their S layer. So you can see that this one is kind of like diamond shaped. It has a lot of little diamonds in it. Depending on what specifically is making up this S layer, you could have different shapes. It doesn't have to be diamond shapes. Some archaea will have like one S layer in their cell wall. Some of them will have like 2 or more S layers in their cell wall. And they could also have other stuff with it, so they might have just an S layer and that is their cell wall, or they might have an S layer and then some other layers additionally. But always, if they have an S layer, 1 or more, the outermost layer is an S layer. Okay, so now we'll go through some of the other external structures of prokaryotic cells. Not all prokaryotes are going to have all of these external structures, so they can have like different combinations of them. The first one is a glycocalyx, which in this image is this really thick kind of blue-green layer. The glycocalyx is made of sugars and proteins. Glyco meaning sugar, so a lot of sugar in the glycocalyx. It's sort of a jelly-like substance that surrounds the cell outside of the cell wall. It's not very strong, but it does protect the cell because since it's gel-like, it's just kind of hard for toxins to get through it. You have 2 different formats of the glycocalyx. It could be a capsule, like in the diagram, or it could be a slime layer, which is more disorganized and slimy. We'll go through each of these in more detail coming up, actually. So that's the glycocalyx. Then you have pili. Pili, you have a lot of different types of pili, and they do different things. So cells would have different combinations of the types, or no pili at all, maybe. Some pili allow cells to transfer genes with other cells, specifically plasmids. Some of them will let the cell stick onto surfaces. Some of them will allow the cell to move along surfaces, and then there's also other things they can do as well. The plural for pili is pilus. Then you have fimbriae, which are little sticks that are sticky, and they allow the cell to interact with surfaces, so sticking onto surfaces. So in this diagram, these short hair-like things that are coming out all over the cell, those are the fimbriae. The plural for that is fimbrium. And this longer thing is a pilus. Then you also have hemi, which are only found in archaea. They're sort of like fimbriae, but for archaea. They also let the cell stick onto surfaces. You have flagella, which are only in bacteria. The flagella is like a long whip thing. So this is a flagella, a flagellum. Flagellum is singular, flagella is plural. The purpose of the flagella is to allow movement. It lets the cell swim, basically. Then in archaea, you have an archella, which is very similar to a flagella or to a flagellum, but it's a little bit different, and it's in archaea. Its purpose is the same. It allows the cell to swim. And those are the main external structures that you'll see in different prokaryotes. So our first external structure is a glycocalyx. It does a lot, well, there's a lot of different ways that the glycocalyx can help protect the cell. So it can help prevent toxins from reaching the cell. It can also help the cell evade the immune system. Sometimes the glycocalyx might have molecules in it that look like molecules that belong to you or belong to whatever host the bacteria bacteria is living in. And anytime the immune system is seeing molecules that look like they belong, then it's less likely to react. And finally, the glycocalyx can help the cell keep its water inside and not dry out. If it's in a very dry place or if it's in a hypertonic solution, the glycocalyx can keep water inside the cell. Another thing the glycocalyx does for cells is just helps them stick to stuff because it can be real sticky. That includes each other. So if you have a lot of bacteria present and they all have a glycocalyx, the glycocalyces of the different cells can all kind of stick and merge together, and that helps form something called a biofilm, which we'll talk, once we go through all these external structures, then we'll come back to the biofilm and talk about it. But it's sort of like, I guess I'd say it's like a tent for bacteria to live inside of. It protects them, and they all live in there in community with each other. You have two basic types of glycocalyces, as I mentioned. You have the capsule and the slime layer. The capsule glycocalyx is more organized and it's well attached to the cell. So here's an example. Actually, all three of these images are showing different types of capsule glycocalyces. And then this image is showing the slime layer glycocalyx. So if the glycocalyx is not as well organized and it doesn't stay attached very well to the cell, then it's a slime layer like this. So you can see little bits of this glycocalyx are getting left behind by the cell here. Especially the slime layer glycocalyx is really useful for forming the biofilm. As you can imagine, it becomes like slime that all these bacteria are all like kind of just suspended inside of, and they're safe there. This is actually a colony of bacteria that are making a glycocalyx, and so you can actually kind of you can almost kind of lift the colony up with a little loop, and that's just showing like how well that glycocalyx is letting the bacteria stick together. Then you have pili, different types of pili. First, you have the type 4 pilus, which will let cells like stick to stuff. Specifically, it lets them stick to host cells. In order for bacteria to get inside of a host cell, first they have to attach to it, and the type 4 pilus will allow them to do that. It also helps them stick to a surface and move along that surface. And it's a special type of motion called twitching motility. So this diagram is showing the path of a cell that's moving by twitching motility. So it's kind of just jumping and jerking all over everywhere. In the twitching motility, you would have these type 4 pili attaching the cell to the surface that it's in or that it's on. And they're kind of under tension. So they would be kind of pulling the cell towards one side or the other. And then if you break one all of a sudden, then the tension is released and the cell will end up shooting off in the opposite direction. And that's the twitch of the twitching motility. Another important type of pilus is the sex pilus or conjugation pilus, which is different from the type 4 because it's hollow on the inside. And it basically forms a little tube that allows genes to travel through it. So you would, if you had this conjugation pilus, you're going to send it out. It's going to attach to another bacterium, and then you can send some of your DNA down through that tube, and the other bacterium will get it. So this is a type of gene transfer between bacteria, or archaea. It's called a sex pilus because when genes are being transferred, it's a little bit like sexual reproduction in a way. So bacteria and archaea have to reproduce asexually, which means that the offspring are genetically identical to the parents. In sexual reproduction, the offspring are different genetically from the parents. So your children have some of your genes, but they also have some of your partner's genes. So they're not identical to either of you because they have a different mix. They have a mix of your genes. So with the sex pilus or conjugation pilus, one bacteria can share some of its genes with another, and then you get whoever has received these genes is now genetically different from their parent bacterium. So it's not sexual reproduction, but it does allow genetic diversity to increase, which is why it's called the sex pilus. And those are just the two most common type of pili. You have others too, yeah, but we won't talk about them. Next we have fimbriae, which basically allow cells to stick to stuff. So that could be a surface, that could also be a host cell. In this image here, you have an E. coli cell that has fimbriae coming off of it. So all these little lines are fimbriae. And then it's surrounded by red blood cells on all sides. And it's actually using these fimbriae to attach to the red blood cells. So if the cell can't attach to its host, then it can't get inside the host, it can't cause disease. The fimbriae will also allow cells to stick to each other, which can help them form the biofilm as well, and just help them live in community with each other. Actually, some cells can send like electrical currents along the fimbriae to other cells, and it's a way that they can communicate with each other. In general, if you had a pilus, you would only have like one. If it's a sex pilus, probably only one. If it's a type 4 pilus, you would have a few. But with fimbriae, you're going to have like a lot all over the surface, sticking out every which way. And then a final thing to note about the fimbriae is that they can be considered a type of pilus. So some scientists would consider that a fimbrium is actually a subtype of pilus. Next, hemi, which are in Archaea, not in bacteria. And they're similar to the fimbriae / the pilus, specifically the type 4 pilus. They're used for sticking to stuff and to other cells and for moving. And the special thing about the hemi that makes them distinct is that they have a hook. So these are images of hemi, and you can see it at the end. It has a hook, almost like a fishing hook. So that really helps it stick to stuff and not let go. And since the hemi let cells stick to each other, it also helps them form a biofilm. Okay, next we have the flagella. You have a lot of types of flagella. Basically, depending on where the flagella are on the cell and how many of them you have on the cell, that's determining the different types of the flagella. So you can have petricous flagella, which would be just flagella all over the cell. So in this image, you have an example of that. These sort of thin lines that are wiggling all over the place, those are flagella that are all attached to this one bacterium. And just coming out all along its surface. So that's petricous. You could also have polar flagella, where the flagella are just at the end of the cell. They could be either at one end, like this has a single flagellum at one end of the cell, or you could have one on each end, which would be like bipolar. You could also have a tuft of flagella at one end or at two ends of the cell. So this bacterium here has a little tuft of flagella coming off of one end. If you have a tuft, there's even another name for that. It's actually two names. One name if you have one tuft, another name if you have two tufts, and we're not going to worry about that. And then finally you have endoflagella, which are only found in spirochetes, which were the type of bacteria that forms a real tight corkscrew. The endoflagella is special because it lies flat along the cell underneath the membrane, actually. Well, sorry, underneath the cell wall / the outer membrane. So these flagella, well, all flagella are allowing the cell to move, but the regular flagella are letting the cell kind of swim. The endoflagella, when they turn, the whole cell is going to turn. And so it lets the cell kind of tumble around. So now we'll go over how flagella work. They basically work by spinning around. Some flagella can spin just one way, some can spin both ways, and the speed can vary for all flagella. And how flagella work to move the cell depends on what type of flagella you have. If you have polar flagella, then overall the movement will be faster and you can have really rapid turns and changes in direction. If you have polar flagella that can turn in both ways, then the cell can just start spinning the flagellum the other way around and it will start moving backwards. If the flagellum will only spin one way, then the cell will need to stop in order to turn and let the kind of environment turn it. It'll kind of drift and turn randomly, and then the flagellum can begin spinning again and it'll take off in a new direction. If the cell has petricous flagella, it's a little more complicated. Movement is slower overall, and it's in a straight line. If the cell wants to turn, then it needs to do something different with its flagella, which we will talk about later on when we talk about more about movement for different microbes. So in order to understand how the flagellum works, we need to go over the specific structure of it. Every flagellum has three parts: the filament, the hook, and the motor. The filament is the main part of the flagella. It's shaped like a spiral. It's made out of the protein called flagellin. So all of this length of the flagellum is the filament. At the base of the flagellum, you have the hook, which is a bit thicker. Its basic role is to connect the filament to the motor. Here's the hook as well. And then the motor has a complicated structure, and it actually makes the flagellum spin. So you can divide the motor also into two different parts. You have the rotator, also called the basal body. Most of the motor is the rotator, would be all of this. Then the stator is the other part that is only at the bottom. It's this ring of proteins that surrounds the rotator. Your structure of the motor is going to differ between gram-positive and gram-negative bacteria. This is for gram-negative bacteria. In gram-negative bacteria, the rotator or basal body has these four different rings in it. So you have the L ring that's embedded in the outer membrane. Then you have the P ring that's embedded in the peptidoglycan of the cell wall. Then you have the MS ring and the C ring that are both embedded in the cell membrane, but the C ring is sticking out into the cytoplasm. Between the MS ring and the C ring, you have flea proteins, which would be these round guys right here. In the center, you have the rod, which rises up from the C ring, passes through all the different rings, and plugs into the hook. Then you have the stator, which has MOT proteins. You have MOT A and MOT B proteins that are surrounding the MS and C rings. If this is a gram-positive bacterium, it's going to be a little bit simpler. Basically, the L ring and the P ring will be missing. So here you have the motor for a gram-positive bacterium. You have the MS ring here, then you have the C ring here, but you don't have the L or P rings. Instead, the rod will just travel through the peptidoglycan of the cell wall and plug into the hook. And it's not shown here, but you would still have the stator proteins, the MOT proteins of the stator, going around the MS and C rings. The role of the stator is to actually cause the rotation to occur. Then the flea proteins are responsible for changing the direction of the flagellum for flagella that are able to change directions. And the other rings have other functions. So we're not going to cover in an incredible amount of detail how this all works, but we will go into a little bit of detail with it, focusing on the part of the motor that is surrounded by the stator, which would be the MS rings and the C rings, right here. So the energy for all of this is coming from the proton motive force. Again, that is a proton concentration gradient where you have protons pumped outside of the cell. So you have more protons outside the cell and less protons inside the cell. The stator proteins, the MOP proteins of the stator, are able to allow protons to travel through them. So here you have some protons traveling through this MOP protein of the stator. They're always going to move from the side where there's more protons to the side where there's less. So that means they'll be moving from outside the cell to inside the cell. This is a type of facilitated diffusion, by the way. When these protons are moving, they're causing conformational changes inside the MOP protein. Those conformational changes, which are just a small change in the shape of the protein, are going to cause certain amino acids in here that have a charge to come into contact with the MS ring and the C ring. And the MS and C rings have charges distributed along them in this kind of repeating diagonal pattern. So if you have charges coming into contact with them, it's going to interact, either giving it a tug or a push. So let's say you had a positively charged amino acid here in the MOP protein that is ending up coming into contact with the MS ring whenever protons move through the MOP protein. So the MS ring has a negative charge right here. So that's going to be attraction between the negative charge on the MS ring and the positive charge on the MOP protein. And that's going to create a little tug. So you're basically going to have little tugs right here. On the other side of the MS ring, the MOP protein over here would also have hydrogen ions going through it. And it'll also have a conformational change that brings its charged plus amino acid into contact with the MS ring and the C ring. But in this case, you have positive charges in that location on the MS and C rings. So that's going to be a little push. So on this side, a little tug, on that side, a little push. Overall, this would be happening all around the MS ring and the C ring, and it's going to cause it to start rotating, or cause these two rings to begin rotating. When they rotate, the rod will also rotate, and that rotation is going to be transmitted up through the hook and into the filament, so the whole flagellum will start spinning. How fast the flagellum spins is going to depend on how quickly you have protons moving through these MOP proteins. So if they're just going kind of slowly, then you have a tug here and there, and you're going to have slow rotation of the flagellum. If the protons are moving through very quickly, then you have a whole bunch of tugs, like in really quick succession, and that's going to create very rapid movement of the flagellum, and correspondingly rapid movement of the cell. And then it isn't shown in this diagram here, but in between the C ring and the MS ring, you would have the flea proteins. If those flea proteins undergo a conformational change, then that's going to switch the direction that the MS and C rings turn when protons are moving through the MOP proteins. Not every flagellum is able to do that, though. So that's flagella. Archaea are similar yet different. So functionally, they're very similar. If you look at kind of a zoomed out view of a flagellum and an archaeum, they look very similar to each other, basically a long whip. But when you zoom in closer, you see that they're actually quite different, and how they work is very different as well. So they are also able to rotate and to spin around. They also can go either in one direction or in both directions, but they're not using the proton motive force in order to power that rotation. Instead, they're using ATP, and the proteins they have in their motor are basically completely different. So you have the Flag-I and Flag-X proteins composing the motor of the archaeum, and these Flag-X proteins are able to use ATP, hydrolyze ATP by breaking off one of the phosphate groups to give that free phosphate and a DP, and then they take the energy from that ATP hydrolysis and use it to spin these Flag-J proteins, which makes the whole filament start spinning. Okay, so that's a basic rundown of the external structures of bacteria and archaea. So now we get to talk a little bit about biofilms. Biofilms are something that bacteria and archaea can form when you have a lot of them together. It doesn't have to be all the same species. It could be multiple species together. The only requirement is that they have to have the glycocalyx. Generally, the slime layer glycocalyx is what helps form the biofilm. So when you get all these bacteria together, or archaea together, and they all have their slime layer glycocalyx, they all will end up maybe attached to a surface, and then their slime layers are going to merge together, and then they'll start growing inside of there, and they're just always completely surrounded by the slime of all their different slime layer glycocalyces, which will become just kind of thicker and thicker. That is very protective for them. It prevents drying out is one thing, but it also just insulates them from their environment. So if you have toxins in the environment, it's hard for those types of things to penetrate the biofilm. It really keeps the bacteria and archaea safe inside of there. And they can all be communicating with each other in there. They can be forming conjugation pili and sharing DNA with each other in there. So you'll find DNA inside the biofilm sometimes. They'll all be sticking to each other, not only with their glycocalyces, but also with their fimbriae or their hemi. And they're all just in a sheltered community. In there, like the biofilm is forming like a tent over them, or maybe like a gated community where they're like sheltered from the outside. So if these are in your body, it's very hard for your immune system to penetrate that biofilm. So they're really well protected against your immune system. Also, any drugs or toxins that are in the environment generally can't penetrate the biofilm. So if you want to destroy those microbes in the biofilm, what you have to do is physically break it up. The most common example of a biofilm that you would be familiar with is actually plaque on your teeth. That's a biofilm formed by the bacteria that live inside your mouth. And the only way for you to get rid of that is by physically removing it by brushing your teeth. But of course, since you don't completely exterminate all the bacteria in your mouth, it regrows again and is there again by the next night. Grody. Okay, so now we'll go through the internal structures of prokaryotic cells, which are pretty similar between bacteria and archaea. You actually have a lot of species that would have specialized structures, but we're not really going to talk about those specialized structures. We'll just talk about the general structures that all cells would have. So first you have the cytoplasm. That's just a word that means all the stuff in the cell. For a eukaryotic cell, the cytoplasm does not include the nucleus. It's everything else in the cell. But for prokaryotes, since they don't have a nucleus, it's just everything inside the cell. Then you also have the cytosol, which is the liquid stuff inside the cell. It's actually more like a gel than a liquid. It's real thick. So that's including basically the water in the cell and anything that is dissolved inside of it. So all your little ions, your glucose and other small nutrients, and also some proteins, some enzymes would be dissolved in the cytosol. So anything that is in solution in the cell would be part of the cytosol. Then you have inclusions also, which are balls of stuff that are not solubilized, so they're not part of the cytosol because they're not dissolved. There's a lot of different things that can be stored in inclusions. Commonly you would see nutrients like fat or starch stored there, but it could be other things too. Different species will have different things that they store in inclusions. So if this is your typical prokaryotic cell, everything that's inside is the cytoplasm. The yellow stuff is the cytosol, and these balls are inclusions. So you have a number of different inclusions. They can be different sizes. Some of them will get really big, and some of them will be small. And they can have different things in them. Then you have the nucleoid instead of a nucleus. So the nucleoid is the part of the prokaryotic cell where you have the chromosome stored. The main DNA inside the cell will be there in the nucleoid. So all this like blue squiggly line, that's all one big circular chromosome. That's the genome of this prokaryotic cell there in the nucleoid. So it's not like super well organized and it's not like separated from the rest of the cell, but it does basically stick to this area and it doesn't move around to other parts of the cell. In addition to that nucleoid, you can have plasmids, like there's a plasmid there, which would be small circles of DNA that are separate from the main genome of the cell and are not stored in the nucleoid. All the little red dots here are ribosomes. Ribosomes, as you probably remember from general biology, are responsible for building proteins using mRNA to build protein. And then finally you have the cytoskeleton. Different prokaryotes will have different formats to their cytoskeleton, but it's basically a framework of proteins inside the cell that helps it maintain its shape and its organization, and it can change over time. So in the diagram here you have different types of cytoskeletons that different types of bacteria or prokaryotes might have. So you could have it just be really simple, a ring of proteins going around the cell, or you could have kind of a spiral going down the cell, where I guess here's a spiral going down the cell, and then this one's more like separate rings going along the length of the cell. This is also part of the cytoskeleton, this kind of curving rod here. So you have different proteins that are making up this cytoskeleton. And the cytoskeleton would be partly responsible for not only the shape of the cell, but for instance, organizing where the nucleoid is at inside the cell. Sometimes the cytoskeleton can also help the cell move, actually. So looking in more detail at the inclusions, it's just a ball of something that's being stored, and it's not solubilized, so it's not dissolved in the cytosol. Some inclusions are surrounded by a membrane, and some are not. And it could be all types of stuff. It's very common to have carbon sources stored in inclusions, so that would include starch or glycogen. It's also common to store phosphate. Microbes that are using sulfur as an energy source will frequently have sulfur inclusions. Another special type of inclusion is a magnetosome. So in this image here, these are bacteria that have magnetosomes, and these little black lines are the actual magnetosomes. So they're storing different minerals in there that are all magnetic, and well, we're not exactly sure what it's for, to be honest, but it probably helps the cell navigate because it lets it basically act as a compass. You can also have something called a gas vesicle inside the cell. It's similar to an inclusion, but it's a little bit different. It stores gas. In order to store gas, you have to have a container for it. So gas vesicles will always be surrounded by a protein shell. It doesn't work actually to have a membrane because gases can penetrate a membrane, so it has to be a protein shell storing the gas, keeping it inside. It's pretty common to find gas vesicles in prokaryotes that live in the water, especially like big bodies of water like the ocean or a lake. And in those large bodies of water, the microbes usually will need to live like at a particular depth. They'll have like one depth that's great for them, and they don't want to be too deep or too shallow. So they can use the gas vesicle to control how deep they are in the water column. It can also help them move through the water column. If the nutrients that they're living off of move to a new depth, they can follow by changing the amount of gas that they have in their gas vesicles. So in this image, you have some gas vesicles in this chain of bacteria living in the water. It would be actually these big kind of balls inside of them are the gas vesicles. So one special structure that can be formed by some bacterial cells is the endospore. It's a dormant form of the cell that is very, very hardy and tough to kill. So basically it's inactive anytime conditions are harsh, meaning there's not enough nutrients, or the environment just isn't a nice place to live. Maybe the temperature is wrong or the pH is wrong, something's off about it. Anytime the going gets tough for any reason, then a cell that could form an endospore will form an endospore, which is basically an inactive form that's very tough and will last through the hard times, basically. Most bacteria cannot make endospores. The ones that can are all related to each other. An example would be the bacteria that cause anthrax can form endospores. So far, we don't know of any archaea that are able to form endospores. The endospore is going to form inside of the regular cell, which is called the vegetative cell because it's growing. It has a lot of different coatings on it that are protecting it from the environment. And it has the genome of the cell, so it has all the DNA from the cell. It also has ribosomes, and then it has molecules to help it be stabilized, basically to stabilize the genome so it's protected. And in addition to that, it has very, very little water in it, so it's quite dry inside of there. So this diagram is showing kind of the spore formation process. The first thing that needs to happen is the cell has to replicate its genome. So you have 2 copies. One of those copies is going to end up inside the endospore. And then the cell is basically just going to start making the coatings that are going to go around the endospore. And there's quite a lot of them. So it just sequentially adds more and more coatings to it. As that's happening, it's also pumping water out from the inside of the endospore. And it's adding all the stabilizing molecules in there, and then it's also going to degrade its other copy of its genome. And when the endospore is finally released, the vegetative cell is going to die. So for that reason, this is not considered a type of reproduction. In reproduction, you have 1 cell, and at the end, you have 2 cells. In endospore formation, you have 1 cell, and at the end, you have 1 cell, which is the endospore. So that's not reproduction because the number of cells does not go up. The endospore is basically just going to sit tight, last out the hard times, and when conditions improve, it's going to germinate and form a new vegetative cell. So we'll go over the structure of the endospore a little bit more with all the coatings that it has. On the outside, it has the exosporium, which is really thick. Inside of the exosporium, it has the spore coat. It can actually have multiple spore coats. Different species can have different numbers of spore coats. Inside of the spore coat, this pale area here is the cortex, which is made out of peptidoglycan. And then inside of that, you have the core. The core has a wall around it, the core wall. Inside of that, you have the cell membrane. And inside of that, you have the contents of the endospore, which would be the genome from the nucleoid, also ribosomes, and then stabilizing molecules. The main stabilizing molecules that you have are the dipicolinic acid, or DPA, and SASPs, or small acid-soluble spore proteins. So the dipicolinic acid, or the DPA, is basically stabilizing the DNA against heat. Normally, heat will denature DNA and destroy it. The DPA is protecting the DNA from that. And the way it does it is by complexing with water. So you don't have very much water inside of the endospore, actually, but you do have a little bit. Any water that you have inside the endospore is going to be complexed with DPA, which means that it's not free, and it can't act as a solvent because it's not free. It's complex to something else. So that means that essentially, functionally, it's like there is no water inside of there, almost. And having less water protects the DNA against heat. So DNA is much more stable. It's better able to resist heat degradation if it's not dissolved in water, basically, if it's dry. The SASPs, the small acid-soluble spore proteins, are also stabilizing the DNA. They also help protect it against heat, but in addition to that, they protect it against UV radiation as well. And then they have one more function, which is to serve as an energy source for whenever the spore is going to germinate. So whenever the endospore is going to germinate and reform a regular vegetative cell, it needs to build a bunch of stuff. And it needs an energy source to do that. And in order to get energy, it's basically going to chew up and eat those the little sasps that are in there. So now we'll cover prokaryotic movement. You have two basic types of movement for prokaryotic cells, swimming and gliding. So swimming we kind of already talked about. Swimming would be, well, like swimming, moving through a liquid medium, so like water. In order to swim, you have to have a flagellum or an archaeal, and that thing is just going to spin around and propel you through the liquid, almost like a boat rotor would do. So that's swimming. You also have gliding, which is moving along a surface instead of moving through something. We've talked a little bit about one way that cells can glide already, actually. So cells can glide using their type 4 pili if they have them, but there's also other ways that they can glide. So there's actually a lot of different mechanisms for gliding. Some cells will have slime that they kind of secrete outside of the cell, and then it just reduces friction and they kind of slide through it. Some cells will have proteins in their membrane that act like tank tracks, basically. So this diagram here is showing that this would be gram-negative bacteria. So here you have the cell membrane, here's the outer membrane, and you have these proteins embedded in those membranes that basically move through the cell membrane. They attach to the surface and then they move through the cell membrane, going basically backwards, so that then the cell will move forwards, similar to how the tracks on a tank work. There's also other types of gliding. There's a lot of diversity actually in the mechanisms that cells use to glide. And not all prokaryotes can move at all. In order to move, you have to have something. You have to have flagella, archaea, you have to have type 4 pili, you have to have one of those other mechanisms of gliding. You have to have something in order to move. So if you don't have any of those things, then you can't move, basically. You're just sitting around until the breeze blows on you or something. In general, Archaea, if they're going to move, they're going to swim using Archaea. They don't glide, at least not that we've found, really. Bacteria would be gliding, and usually either rods or bacilli or filamentous bacteria are able to glide by different mechanisms. You can always tell if cells are able to glide when you culture them by looking at the shape of the colonies that they form. So here you have images of different colonies. These colonies are formed by a species that does not glide, and they look like little circles. This colony is formed by a species that does glide. So it's got all these striations on it, which would be the bacteria kind of moving out from the center of the colony. Also, if you watch a gliding colony over time, you're going to see it changing, not just growing and getting bigger, but moving out, and kind of these striations are going to change where they're at on the colony surface. Related to movement, we have taxis. Taxis is like movement with a goal, basically. If you're moving to get to somewhere or away from somewhere, that's called taxis. And the plural of that is taxis. You have a lot of different types of taxis actually, but the two common ones defined in prokaryotes are chemotaxis and phototaxis. Chemotaxis is the most common by far actually. In chemotaxis, the stimulant that you're moving to or away from is a chemical of some type. So that could be a nutrient. If you kind of sense that there's nutrients in a certain direction and you move towards them, that's chemotaxis. Or maybe it's a toxin that you're trying to stay away from, and that's also chemotaxis. In phototaxis, you're moving to or away from light. Usually you would only find that in prokaryotes who are photosynthetic. So they have a use for light, so they would be moving to light. But there's also exceptions, and you can find it in other types of microbes as well. So you have a couple of types of phototaxis. You have scotophobotaxis, which is moving from dark areas to light areas in general. and then you have true phototaxis, which is moving from low light to high light. So if you're doing scotophobotaxis, then basically as long as you're in the light at all, then you're fine there. You're going to stay put. If you're in the dark, you're going to move to wherever there's light. And once you find that light, you're staying put. If you're doing true phototaxis, then once you get to the light area, if there's another area that's in more light, you're going to move to that other area that has the highest light. So this diagram at the bottom is showing chemotaxis. A way that you can test for chemotaxis is just to have your microbes growing in some medium, liquid medium, and then stick a tube inside of the medium that has an open end and has some type of chemical inside of it in solution. So if you stick a tube that has a solution in it and an open end into this flask that has bacteria growing in it, and you wait, some of them are going to end up going inside of the tube just, randomly. And the chemicals that are in this tube are going to start diffusing out into the flask. So you're going to have a concentration gradient where the edges of the flask have very little of that chemical, and then as you get closer and closer to the opening of the tube, then you have more and more of the chemical. If these bacteria are not attracted to the chemical or repelled by it, so if they don't undergo taxis with reference to this chemical stimulant, then you're just going to have the same, ultimately you're going to have the same concentration of bacteria inside the tube as inside the rest of the flask. If this chemical is an attractant for them, then they're going to move towards it, and as time passes, you're going to see that they are really crowding into that tube, and around the opening of the tube, you have a bunch of bacteria trying to get in. So they've sensed this chemical is there, and they like it, and they're trying to move towards it, and they end up, being more concentrated where the chemical is more concentrated. So this would be chemotaxis towards the stimulant. If the chemical is a repellent for them, then they're going to move away. So as time passes, you're going to end up having bacteria concentrated away from the tube. You might have a couple in the tube still just by random chance, but the tube should be relatively free of bacteria. Also, the opening of the tube should be relatively free of bacteria, and you're going to have more bacteria in the areas of the flask where you have less of that chemical. So this is also chemotaxis, but in this case, the stimulant is a repellent, and they're moving away from it. The top image is showing bacteria that are undergoing phototaxis, so they're moving in response to light. In this case, the light is kind of shining onto this strip of paper, and it's broken up by a prism into the different wavelengths, so it's not just like white light here. Photosynthetic bacteria will have like certain wavelengths of light that allow them to do photosynthesis, and other wavelengths will not help them. They can't do photosynthesis with it. So they're actually generally going to be able to sense the specific wavelengths of light that they can use for photosynthesis. And if they undergo phototaxis, they're going to move to wherever they're sensing the most quantity of that wavelength that they can use for photosynthesis. So in this case, these bacteria are concentrated here. They've undergone phototaxis, and they're concentrated here at 400 nanometers, at 600 nanometers, and around 850 nanometers. So they have migrated actually to wherever there's the most light that is useful for them for photosynthesis. So in order to do chemotaxis or phototaxis or any other type of taxis actually, the one thing that the microbe has to do is be able to sense that stimulant. So if you're going to do phototaxis, you have to be able to sense light. You have to have a receptor that lets you sense light. If you're going to do chemotaxis in reference to a specific stimulant, you have to have a receptor for that stimulant that lets you identify it. Here's an example of taxis for bacteria that have Petricus flagella. And in this case, they are moving towards this stimulant, which would be a nutrient for them. So Petricus bacteria are going to move using runs and tumbles. A run is movement forward in a straight line. The flagella, for Petricus bacteria, the flagella will be sticking out all over the surface of the cell. When they're doing a run, the flagella are all going to rotate in synchrony with each other, and they're all going to be turning in the counterclockwise direction. And that's going to cause the bacteria to move forward. Then at some point they're going to stop doing that run and they're going to do a tumble instead. In the tumble, they reverse the direction that the flagella are turning, so they're going to turn clockwise now, and they're going to do it asynchronously, so basically just at different times. So here you have a bacterium doing a run, then it stops. It does a tumble. When it does the tumble, since the flagella are moving backwards and they're asynchronized, they're going to spread out from each other, and the cell is going to rotate randomly. Once it's in a new direction, then the cell is going to do another run and then another tumble. So these bacteria with Petricus flagella are always moving by doing a run and then a tumble to reorient themselves and then another run. And this is just random. They're going around randomly. But if they're doing chemotaxis, then their movement is not random. They're moving towards or away from something. They're going to do that by changing the frequency with which they're doing runs and tumbles. So in this diagram here, you have a stimulant in the center, which is some type of nutrient that these bacteria are attracted to. Each of these black dots is a bacterium with Petricus flagella, and then the line it's leaving is just showing the path that it has taken. Every time the line is straight, the cell is doing a run. Every time the line bends, the cell is doing a tumble. In order to navigate to this stimulant in the center by chemotaxis, the cells are going to be sensing how much of that chemical stimulant is around. So you have a lot of it right here in the center, but it's also going to be spreading out from there by diffusion in a concentration gradient. So you can see that gradient here, where close to this dot, it's kind of a darker color, and if you go farther away from the dot, it's a lighter color. That's a concentration gradient where you have only a little bit of the nutrient out here at the edges, and you have a lot more of it in towards the center. These bacteria are able to sense that. So if they sense that the concentration of the nutrient is going up, they're going to do more runs. They figured they're going in a good way, the direction is right, the place is good, time to run to the place. They're going to do less tumbles. Every time they do a tumble, they change their direction. If they are sensing that there's less of this nutrient around, then they're going to stop running, they're going to do a tumble. Right? Because that means that now they're going in a wrong direction. If the concentration of the nutrient is going down, now they're not going the right way anymore. It's time for a tumble, reorient, hopefully go in a better way next time. This is called a biased random walk, because they're not actually able to say, like, the center is here, the center is in this direction, I'm just going to run that direction. They can't do that. They can't pick their direction. It's just random. But it's biased because they're going to run more if they're going the right way, and they're going to tumble more if they're going the wrong way. And eventually they end up getting to the center. And it's basically the same if the stimulant is bad. If the stimulant was a bad thing that they didn't want to go towards, then as they get closer to it, they would tumble more. And as they get further from it, they would run more. And so they would end up running away from it instead of falling into it. So that's an example of how chemotaxis can work. Okay, so now we get to go over the remaining domain of life, Eukarya and the Eukaryotes. And again, we'll go over the different structures that they have and their basic functions. Starting with the cell membrane again, it's basically very similar to the cell membrane for prokaryotes. One thing that I'll mention that we didn't talk about for prokaryotes is that eukaryotes have sterols in their cell membrane. Cholesterol is the main example of a sterol that we're all familiar with. And we actually have cholesterol embedded in the membranes of all of our cells. So that's where our cholesterol is coming from, really. Eukaryotes will have sterols, frequently cholesterol, but different eukaryotes will have different sterols. So like fungi will have a different type of sterol in their cell membrane. Bacteria and archaea can also have something like similar to a sterol embedded in their cell membrane, but not actually a sterol. The sterol is basically giving the membrane greater strength and letting it resist temperature better, specifically low temperatures. So low temperatures can kind of make membranes freeze. It can make them like very brittle, and that makes them break easily. If you have sterols in your membrane though, then as the temperature goes lower and lower, your membrane stays fluid. It doesn't freeze or get brittle. So the lower the temperature is, probably the more sterols would be in the membrane for a cell living at that temperature, if it's a eukaryotic cell. Eukaryotes can also have cell walls. A lot of them don't, and a lot of them do. Plants will have cell walls, usually made out of cellulose, which is a polysaccharide. Actually, all of these basically are polysaccharides. Almost all of them are polysaccharides. Algae will have a cell wall. It could be made out of different things. It could be made out of agar or carrageenan. Those are both polysaccharides. Could also be made out of silica, which is not a polysaccharide. It's basically similar to glass and other things as well. In this image here, these would be different algal cells that all have silica making up their cell walls. And you can see their cell walls have all these like crazy shapes, and they're very pretty because it's like glass. Fungi also have cell walls. They can be made out of cellulose like plants. They could also be made out of chitin, which is another polysaccharide. Animal cell walls, animals don't have cell walls. Protists also don't have cell walls. So your plants, algae, and fungi have cell walls. Animals and protists do not. The cell wall is having the same function for eukaryotes as it does for prokaryotes. The only exception would be for multicelled eukaryotes like plants. The cell wall will also help stick the cells together to make the structure of the larger organism. But of course, plants are not microbes. Eukaryotes do not have as many external structures as prokaryotes do, but they can have glycocalyces and a couple other things. So in general, eukaryotes are either going to have a cell wall or a glycocalyx, but not both. So if you have a cell wall and you're a eukaryote, you do not have a glycocalyx. If you do not have a cell wall and you're a eukaryote, then you might have a glycocalyx, or you might have nothing at all. So that means that the main microbial type of eukaryote that could have a glycocalyx would be a protist. Your fungi and algae are going to have cell walls instead. Eukaryotic glycocalyces are a bit different from prokaryotic. They are not as organized and they don't have the same function, so they're not really for protection. Eukaryotic cells have other ways of protecting themselves from the environment, and the glycocalyx is more likely to be used for anchoring the cell to a surface or for communicating with other cells. So this image is an example of glycocalyces within your own body, actually. This is a blood vessel, and all blood vessels are lined with the endothelium, which is just a single layer of flat cells. And the endothelial cells have a glycocalyx around them, and it's mostly sticking out on the side of the cell that faces into the blood vessel and would be in contact with the blood. So this like all these kind of hair-like or spiky structures, that's the glycocalyx or glycocalyces of these different endothelial cells underneath there. The glycocalyx in this case is helping regulate the function of the blood vessel. So it's regulating like kind of how sticky the walls of the blood vessel is or are, and like how clotting occurs and stuff like that. So eukaryotes can also have flagella, but their flagella are different from prokaryotic flagella in their distribution, their structure, and their function. Eukaryotic flagella are located only at one end of the cell. It can either be a single flagellum or it could be a tuft. So not at both ends of the cell and not all over the cell. They have a different structure than the prokaryotic flagella. They're much thicker. So here's a prokaryotic flagellum, and here's a eukaryotic flagellum, and much thicker as you see. The prokaryotic flagellum is made out of the protein flagellum. The eukaryotic flagellum is made out of these microtubules, which are like long filaments. Those microtubules are themselves made out of the protein tubulin. The function is also different. It's still letting the cell move by letting the cell swim through its liquid medium, but it doesn't rotate like the prokaryotic flagellum. Instead, it whips back and forth in these little kind of waves. So that means that it's going to create a slower, steadier type of movement. You don't have like real fast runs and tumbles and stuff. Eukaryotic cells can also have cilia on them, which look kind of like fimbriae, but they are not fimbriae. So similar to fimbriae, they're small, kind of hair-like little sticky sticks. Well, but not sticky, just sticks. And they cover the whole surface of the cell like fimbriae do. But unlike fimbriae, they're not used for attaching to stuff, and they're able to move. So cilia will whip back and forth like this, sort of similar to the eukaryotic flagellum. The cilia can be used to move the whole cell, but they could also be used to move stuff along the cell surface. For microbes that have cilia, they would generally be used to move the cell. Usually it would be multicelled eukaryotes that use cilia to move things along a cell surface. Like for instance, in your respiratory tract, the cells have cilia so that when you breathe particles in, they can be moved up and out. Cilia are also made out of microtubules, same as eukaryotic flagella. And in both cases, the protein dynein is being used to make the microtubules slide past each other, which causes the whip-like movement of the flagella or the cilia. And again, not all eukaryotes have these structures. They could have one, so they could have flagella, or they could have cilia, or they could have both, or they could have none. Next, we have the internal structures of eukaryotes. So first, some structures that are basically the same as what you have in prokaryotic cells. So you have the cytoplasm. The difference there is that in eukaryotes, the cytoplasm does not include the nucleus. So it's everything inside of the cell except for the nucleus. You also have the cytosol, which is just the same. It's the water inside the cell and everything that is dissolved inside of it. You have the cytoskeleton that functions the same as the prokaryotic cytoskeleton, so it gives the cell shape and organization, but it has a different structure from prokaryotic cells, and it also has an extra function, or maybe two extra functions. So it helps the cell transport things inside of it. If the cell needs to move stuff from one side of the cell to the other side, it would use the cytoskeleton to do that. For some eukaryotic cells, the cytoskeleton can also move the entire cell. Then you have ribosomes, which work the same way as prokaryotic ribosomes, but they do have a different structure. And in your prokaryotes, the ribosomes are just floating in the cell. In your eukaryotes, some of the ribosomes are floating, and some of them are attached to an organelle called the rough endoplasmic reticulum, or to the nucleus. Then in addition to these structures, you also have the membrane-bound organelles, which you do not find in prokaryotes. So the cytoskeleton we'll talk about in a bit more detail. It has a lot of functions in eukaryotic cells, maintaining shape, organization, and helping the cell move stuff around, or potentially move the entire cell. You have three different types of filaments that make up the cytoskeleton, all made out of different proteins. So you have microtubules, the same as what you would find making the flagella or the cilia, made out of tubulin. You also have microfilaments that are made out of actin, and you have intermediate filaments that are made out of keratin. Keratin is also the protein that is filling up your skin cells and builds your nails and your hair. Then you have some motor proteins in there as well. You have myosin and you have kinesin. So myosin is found on the microfilaments. So you have actin in the microfilaments building the filament, and then you have myosin that can walk along the microfilament basically. That basically allows the microfilaments to slide past each other and kind of change their shape and organization. So the cytoskeleton can change its overall shape, and when that happens, the cell could change its overall shape, and it would be myosin on the microfilaments that's allowing that to happen. Then you have kinesin on the microtubules. So kinesin is going to be used to move stuff around in the cell. And in this diagram here, it's showing that. So this is a microtubule made out of little tubulins. And this is your kinesin protein. And here's a vesicle that's filled with stuff that needs to be moved to a new place. And the kinesin protein grabs the vesicle on one end. On the other end, it has like a couple of feet that's going to use ATP to walk along the microtubule with. And it just follows the microtubule as though it's like a road or a railroad track, basically, to get from point A to point B. And then you have your membrane-bound organelles. An organelle is basically a small organ, as what the name really means, little organ. It's a compartment in the cell that's usually surrounded by a membrane, and it has a specific function. So inside of that organelle, you're going to have all the stuff you need for that function. Most organelles would be surrounded by a membrane, but not all of them are. So you can consider the cytoskeleton to be an organelle, actually, even though it doesn't have a membrane. Most of your organelles are in eukaryotes, especially the membrane-bound organelles are basically in eukaryotes, and the nucleus is a type of membrane-bound organelle. Of course, prokaryotes also have a cytoskeleton, and that is an organelle, but it's not a membrane-bound organelle. Organelles have a lot of benefits, especially the membrane-bound organelles, because they are actually establishing a separate environment from the rest of the cell. So it lets you get all the stuff you need for a specific task and put it there in that organelle. So everything you need is like right there on hand and easy to access. It also lets you separate things that don't go together, like making and breaking. So you can have, like, the cell has a need to degrade stuff sometimes if it gets old and just needs to be recycled, or if it's like broken, it needs to be recycled. So the cell needs to be able to degrade proteins, but it also needs to make proteins and have proteins that are not being degraded. So in order to separate those functions of making and breaking, the cell will use organelles. So it'll have one organelle that is for breaking down protein. All the stuff you need to break down protein will be right there, and it's not in the rest of the cell, which means that the rest of your proteins that are not inside of that organelle do not get degraded. There's another benefit for eukaryotic cells, since they have such a big volume. That means that it's hard to get a high concentration of reactants if you don't use membrane-bound organelles. In order for a reaction to go quickly, a lot of the time you need to have a high concentration of reactants. And if you didn't have membrane-bound organelles, that would mean that you need to fill the whole cell with a lot of that reactant. So you would need a lot of reactant to fill up the whole volume of the cell. With membrane-bound organelles, if you take those reactions and those reactants and stick them inside the organelle, you don't need as much reactant to get a high concentration so that the task can proceed quickly. So it's very helpful there. Also, in general, the space inside an organelle is the lumen. Some organelles will have like a special name for the space inside of them, but in general, it's the lumen. So first, we'll start with the nucleus, which holds the DNA of the cell, holds the genome of the cell. In eukaryotes, the DNA is packaged in a lot of ways. Actually, it's pretty complicated, but the first layer of packaging is always wrapping the DNA around these bundles of histone proteins. So here you have histone proteins with DNA wrapped around them, and then you have one other histone just on the outside, like kind of anchoring the wrapped DNA in place. So you get these little beads, basically. It's called beads on a string. The histone proteins with the DNA wrapped around them form a bead, and then the DNA itself is the string. This helps pack the DNA so you can have more of it in a smaller location, in a smaller space. When the DNA is packaged with histones like this, it's called chromatin. And you have a couple different types of chromatin. You have euchromatin and you have heterochromatin. Euchromatin is loosely packed and heterochromatin is tightly packed. So this is the first level of packing, but there's more packing on top of this. You would take these histone kind of beads and wrap them all up to make like a filament, and then you would wrap that up to make a fiber. It gets kind of complicated. So if your DNA is not very packed, if it's more like just around these histones, beads on a string, and that's it, then that is euchromatin. If it's tightly packed, then that's heterochromatin. And the most tightest packed DNA is an actual like chromosome in the way that you're used to seeing it, like with the little arms, 2 on the top and 2 on the bottom. And so that's like the most densely packed the DNA can be in heterochromatin. Euchromatin is active, which means that you can actually use that DNA to build protein. Heterochromatin is inactive, which means that you cannot use the DNA to build protein. The reason is it's just too tightly packed. In order to use that DNA to build protein, you have to make mRNA from it. That means you have to get enzymes up in there. You have to get the RNA polymerase in there, and a variety of other enzymes have to go inside, have to get up in there. So you would have to move those histones out of the way. But if the DNA is really tightly packed, there's just no space. There's no space for all those enzymes to go. So you can't use the DNA, basically. In this diagram of the nucleus, your euchromatin is in yellow, and the heterochromatin is in green. So you actually have a lot of DNA that's in heterochromatin, but because it's very tightly packed, it doesn't take up as much space. Euchromatin takes up a lot more space, and it is active DNA that can be used to make protein. Then you also have this smaller ball inside of there. It looks kind of like a yolk in an egg. That's the nucleolus. The nucleolus is a special place within the nucleus where you're building ribosomes. So you have rRNA that's built there, and then you have the proteins for the ribosomes that are brought in from the rest of the cell, from the cytoplasm into the nucleus, into the nucleolus, and they're assembled together with the rRNA to build a ribosome. And then you transport that out of the nucleus. The nucleus is unusual partly because it has 2 membranes around it. Most organelles have only 1 membrane, but the nucleus has 2. So here's the outer one, and then the inner one you can see right here. Those 2 membranes together are called the nuclear envelope. And then you have these dots in there, which are nuclear pores that allow stuff to go in and out. And then these little pinkish dots are the ribosomes that stud the nucleus, actually. Next, you have mitochondria. That's also an organelle that has 2 membranes. Its job is to make ATP. The singular version of mitochondria is mitochondrion. So you have this outer membrane that has a lot of pores in it. And then you also have the inner membrane that is folded up to form cristae. So these little folds are the cristae. Then inside the inner membrane, you have what would be called the lumen, but in this case we have a special name for it, which is the mitochondrial matrix. Inside the matrix and the cristae is where you're actually making ATP. So here's some other views of it. This would be the matrix and the cristae. Mitochondria are really unusual organelles because they have their own genome. So they have genes on chromosomes that are actually inside of the mitochondrion and not stored in the nucleus with all the other DNA. And basically all eukaryotic cells would have a mitochondria, but there are some exceptions for eukaryotic cells that are anaerobic, which means that they don't use oxygen. If they don't use oxygen, then they don't have a need for mitochondria, because mitochondria make ATP in a process that relies on oxygen, which we're going to go over in chapter 3. You also have hydrogenosomes in some eukaryotes. It's not very common actually to find hydrogenosomes. They're similar to mitochondria. They actually probably came from mitochondria by evolution, but they are now different. They don't have cristae, and they cannot make ATP in the same process that mitochondria use that relies on oxygen. Instead, they conduct fermentation. So anaerobic eukaryotic microbes can have hydrogenosomes that they use for fermentation. And these would be examples of hydrogenosomes. And here is a hydrogenosome. Well, actually, here is the hydrogenosome, and it is carrying out fermentation. So we're going to actually go over in chapter 3 again, fermentation and this whole thing. But starting with glucose, cells can do a process called glycolysis to make pyruvate. Once you have pyruvate, if you're aerobic, then you would send that to the mitochondria to make ATP using oxygen. But if you are not aerobic, if you're anaerobic, then you're going to do fermentation with the pyruvate instead. And that would have different products actually, and it makes a little bit of ATP. That could be occurring inside of the hydrogenosome. Then we have chloroplasts, which are also unusual. They have 3 membranes actually. They make ATP, and they also make sugar. They are the site of photosynthesis for photosynthetic eukaryotes. They have kind of a complicated structure. The first 2 membranes that they have are just surrounding the outside of the chloroplast. And then the innermost membrane, the 3rd membrane, has a bunch of folds in it. It folds to make these little discs called thylakoids, and then you can have those thylakoids stacked up. A stack of thylakoids is a granum, plural grana. It's the grana where you're actually making ATP in photosynthesis. It's the grana that are capturing light and producing ATP. So this would be a cell that has chloroplasts, and these red dots here are the actual chloroplasts. Usually they're green, but they don't have to be. These are more cells with chloroplasts, and here they are actually green because of chlorophyll. This is the inside of a chloroplast this is a granum actually. So this here is a thylakoid. This is another thylakoid. So all these little layers are thylakoids, and the whole stack is a granum. And it would be in here in these membranes where you're capturing light and making ATP in photosynthesis. Then in the next part of photosynthesis, you make sugar, usually glucose, and that would be occurring in the stroma, which is the space outside of those grana, but within the within the other 2 membranes of the chloroplast, the internal and external membranes of the chloroplast. So this here is the stroma. Chloroplasts are similar to mitochondria because they have their own genome. So they have their own genes that are stored inside the chloroplast and not inside the nucleus, which is very unusual. It's only seen in mitochondria and chloroplasts, and sometimes hydrogenosomes that probably came from mitochondria. Chloroplasts would only be found in eukaryotes that do photosynthesis, of course. So that would be plants and algae, basically. The only microbial eukaryotes that have chloroplasts would be algae. Even though the chloroplast produces ATP, those organisms would still have mitochondria, and they would be using their mitochondria to make most of their ATP. Most of the ATP that is made in photosynthesis is just used to make the sugar, actually. So that brings us to endosymbiotic theory, which is about the origin of mitochondria and chloroplasts. This theory says that mitochondria and chloroplasts used to be independently living bacteria. They were engulfed by a larger eukaryotic cell, an early eukaryote, and instead of dying there, they found it to be a nice place to live. They divided and reproduced and were kind of passed on to the daughter cells of this eukaryote when it divided, and eventually became organelles instead of organisms capable of living independently. This symbiotic relationship between these bacteria and the eukaryotic host cell would have been mutually beneficial. The host cell would be getting energy, either it would be getting ATP from early mitochondria, proto-mitochondria, or it would be getting sugar with a little bit of ATP from protochloroplasts. And the bacteria would also benefit. They would be getting a stable environment. So they wouldn't need to worry about what happens if the pH gets out of whack, what happens if the temperature changes, what happens if there's not nutrients or if there's toxins. None of those things will happen when you're inside of the eukaryotic cell. The eukaryotic cell is maintaining a stable internal environment, which is a nice place to live. So this theory was first proposed at the end of the 1800s, and basically people thought it was crazy, so they just dismissed it. And there wasn't really very much evidence for it at that time. But in the 1970s, that's when DNA was discovered in mitochondria and chloroplasts. So that discovery led the scientist Lynn Margulis to basically investigate this idea for her dissertation. She was advised to not do that because, because it was considered to be a crazy idea, basically. But she decided that with this new evidence that DNA exists in mitochondria and chloroplasts, that makes the theory much more theory-like, much more reasonable, and she thought she could get more evidence for it, and she did. So now, because of her work, it's actually a widely accepted theory. So this diagram is showing how that might kind of have worked. So you have here a prokaryotic cell that has a nucleoid, which would have evolved to be a eukaryote with a nucleus. This part, that's not part of endosymbiotic theory. Endosymbiotic theory does not say where eukaryotes come from, or how they got a nucleus from a nucleoid. But starting with this early eukaryote, the theory says that a bacterium entered the eukaryotic cell. It would have been a proteobacterium and prospered there, divided, and eventually evolved to be a mitochondrion, which was supplying the cell with ATP, where the host cell was supplying the mitochondrion with a stable environment. And then along a different lineage of the early eukaryotic cells that had already gotten these mitochondria, you would have had another bacteria enter the cell, this time a cyanobacterium, which again prospered there instead of dying and divided and eventually became a chloroplast. So there's a lot of evidence to support this theory. First off, there's the fact that mitochondria and chloroplasts have two membranes surrounding them, which is not normal for organelles to have. But that is what you would expect if a bacterium was engulfed by a eukaryotic cell. That would have been by endocytosis. So you might remember endocytosis from Intro to Biology, but it's basically the way that cells ingest large particles. They're going to have their membrane, their cell membrane, invaginate to form a little depression. The particle they're trying to ingest will fit into that depression, and then the membrane will pinch off or pinch closed at the top, and you end up with this particle inside of a vesicle. So if this particle was actually a bacterial cell, then it has its own cell membrane, and then it ends up inside the vesicle, which is another membrane. So now it has two membranes. That's a double membrane. So that could explain why these organelles have two membranes. These organelles also have their own DNA, and it doesn't look like eukaryotic DNA actually. It looks like prokaryotic DNA. Eukaryotic DNA is a linear molecule. Eukaryotic cells have linear chromosomes. Bacterial cells have circular chromosomes. And the DNA inside of chloroplasts and mitochondria is a circular chromosome. So it's more like bacterial DNA. And it also has other similarities to bacterial DNA as well. They don't have enough DNA actually for all of their stuff. So Some mitochondrial proteins are encoded by the DNA inside of the mitochondrion, but some of those proteins are encoded by genes that are actually now in the nucleus of the cell. And the same thing for chloroplasts. Some chloroplast proteins have their genes inside the chloroplast. Some of them have their genes inside the nucleus. So some genes would have, if this theory is correct, some genes would have had to start off inside the mitochondrion or inside the chloroplast and then would need to migrate into the eukaryotic nucleus. Another piece of evidence is that mitochondria and chloroplasts have their own ribosomes. No other organelle has its own ribosomes. Also, these organelles divide independently from the rest of the cell. So most organelles inside the cell divide whenever the cell divides, but mitochondria and chloroplasts are kind of dividing on their own schedule. Also, if you look at their rRNA, it doesn't look like eukaryotic rRNA. It looks like bacterial rRNA. And you can analyze their DNA and discover which bacterial groupings they are related to. So for chloroplasts, that's cyanobacteria, and for mitochondria, that's proteobacteria. So if there's no actual relationship between mitochondria and proteobacteria and between chloroplasts and cyanobacteria, then it's really difficult to understand why they would have the same genetic sequences. So this theory is very widely accepted, but it's not universally accepted. There are some people who don't believe that this is what occurred. One of the main things they point to is the fact that some genes from the mitochondria and from the chloroplast have moved into the nucleus, which would be difficult to do. There's not really a mechanism that can explain like how that would have happened. It's not really easy to see how that would have happened. For me, the DNA moved one way or another, right? Either it went from the mitochondria to the nucleus, or it went from the nucleus to the mitochondria, and either way is hard to see how it would have happened, but it did happen somehow. And that's into symbiotic theory. So now we'll go through the other membrane-bound organelles inside of eukaryotic cells. First, we have the endoplasmic reticulum, which you have two types of. You have the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The endoplasmic reticulum is located around the nucleus, and it's these layers of membrane. So the membrane of the endoplasmic reticulum is folded to make all these layers. The layers are all connected to each other, and the membrane itself is connected to the nuclear envelope at some of these nuclear pores. The rough ER is called that because it has ribosomes studding its surface. It is involved with modifying proteins, folding proteins, and then transporting them out in vesicles. The smooth ER does not have ribosomes on it, which is why it's smooth. And it does not have anything to do with making proteins. Instead, it makes lipids and transports them in vesicles. It can also store calcium. For most eukaryotic cells, that's not a very important function, but for some, it is. Then you have the Golgi apparatus. That's also called the Golgi body. That's also layers of membrane, but they are not connected. So here's the Golgi apparatus, and the layers of membrane are not connected to each other. So they're separate compartments, basically. The Golgi apparatus is involved with modifying and sorting proteins. It's going to be receiving vesicles from the rough endoplasmic reticulum. Those vesicles are going to contain proteins that are just made by those ribosomes, folded in the rough ER, may be modified there and sent into a vesicle that goes to the Golgi apparatus. When the Golgi apparatus receives these vesicles, it's going to start modifying the proteins. It modifies them in different ways depending on where they need to go. Then at the end, so the protein is going to be the protein is going to be traveling through these different layers of the Golgi apparatus. In each layer, it gets modified in a different way, and it might skip some layers also, depending on what type of protein it is and where it needs to go. And at the end, when it comes out the other side, it's going to go into a vesicle that will be sent to its final destination. So some of these proteins will be going to the cell surface, even outside the cell maybe, but some of them will be going to other locations inside the cell. Not every eukaryote has a Golgi apparatus. Most eukaryotes have it, but some don't. So the main theme of function for the endoplasmic reticulum, for the rough endoplasmic reticulum, is folding proteins, and the main theme for the Golgi apparatus is sorting proteins. But both organelles also modify the proteins to send them to different places. The last membrane-bound organelle that we're going to talk about is the lysosome. There's also others, but we're just going to talk about the lysosome. The others are less common or just found in some eukaryotes, but not others. The lysosome is the main digestive organelle of eukaryotes. So it has digestive enzymes, and it's very acidic. It has a low pH. If the cell has taken in some nutrients from the environment and they're kind of big, they need to be broken down, it's going to put them in a lysosome to break them down. But also cellular structures that are older or broken proteins, anything that's old or broken inside the cell is going to be put inside a lysosome to be broken down into its components that can then be recycled. So here's an example of a lysosome, and these globs inside of it would be digestive enzymes. Most of them are designed to break down proteins, and then the low pH inside of here helps that process occur. And this image is related to the formation of the lysosome. So you're going to have some proteins that are digestive enzymes. They're made by ribosomes, folded in the rough ER, then sent in vesicles to the Golgi apparatus. That's going to modify them so that they're sorted to the correct location. They're going to exit in this vesicle, which ultimately is going to be fused with another vesicle that contains the stuff that needs to be broken down. After that fusion has occurred, we call that a lysosome. So in this image, the stuff that needs to be broken down has come from outside the cell. It's food from outside the cell, but it could also have come from inside the cell.